Hi, I'm David Johnson, Senior Director of DMPK at BioAgilytix. We are a leading bioanalytically focused CRO, and we are able to provide you with bioanalysis for every phase, every modality in support of your drug discovery program and drug development program as well. We focus on pharmacokinetics, immunogenicity, biomarkers, and CMC analytical. And within these areas, we provide assay development, validation, and sample testing. Today, we're going to talk about total ADME support for oligonucleotides. And in preparation for that talk, I want to give you a little bit of background on the importance of ADME in drug development. ADME stands for absorption, distribution, metabolism, and excretion. It basically describes how a drug enters, is processed by, and leaves the body. ADME data are critical for dose and route selection, and for this reason, ADME data are required in regulatory filings. Some examples of ADME data are for absorption, measuring the extent and rate of the drug entering circulation, as well as kinetic parameters, bioavailability, and bioequivalence. For distribution, some of the parameters are protein binding and blood partitioning. For tissue concentrations, those are also included in distribution. Metabolism is involved with the clearance of compound by conversion into metabolites, or we're wanting to look at possible metabolic pathways for that conversion. We want to look at pre-systemic metabolism, first pass effect, key metabolic enzymes, similarities, differences in toxicology species. And then for excretion, we're looking at routes and extent of excretion. These ADME data are collected for preclinical phases as well as phase 1 through 3 in clinical trials. Most preclinical ADME assays can be conducted using a cold compound with LCMS or another platform depending on the modality. QWBA does require radiolabeled compound at the preclinical phase. For phase 1 through 3, also most work is done with cold compound. However, hot compound is needed for definitive absorption, metabolism, and excretion study at the human state. So we need a way to produce these hot compounds. For small molecules, the synthesis of these is not necessarily trivial, but organic synthetic techniques are well established for this modality. Oligonucleotides, on the other hand, for the synthesis can be tricky. Multiple sites may need to be radiolabeled so that we can cover a range of different shortmers. Yields can be low. And those are a little bit trickier. Sekisui Medical, one of our partners, has pioneered a novel radiolabeling technology to address the challenges of performing radiolabeled ADME studies with oligonucleotides. And I would like to introduce Takahiro from Sekisui to further discuss their process, and I'd like to hand it over to him right now.
I'm very appreciated to have an opportunity to present our efforts for ADME study in this webinar, and thanks for great support for BioAgilytix members. My name is Takahiro Kikuchi from Japanese CRO Sekisui Medical. Today, I'd like to introduce our facility and our ADME service through our example study. This slide shows compliance of our facility. In left picture, we are audited by PMDA every 3 years as a GLP-compliant facility for TK analysis. The last audit was in 2022, and we have been certified by them. In middle picture, we renewed full accreditation by ALAC International in 2023. This means we are animal-friendly lab and a good welfare. In right picture, our facility has been licensed to use radioisotopes since 1965. This means we have a lot of experience using radioisotopes. We provide service for many parts of the drug development process. In the preclinical stage, we perform radiolabeled custom synthesis, screening, PK studies, and pharmacological studies. Recently, our service has been expanded into field of the clinical hot study using radiolabeled compounds such as human mass balance study and absolutely viability study. In this webinar, we will introduce an example service using a novel labeling technology for nucleic acid focused on RI custom synthesis and in vivo pharmacokinetic study. Before introducing our example effort, let me briefly explain ADME. ADME is method of pharmacokinetic evaluation using radioisotope labeling compounds. In case of using unlabeled compound in ADME study, it is difficult to quantify the total amount including metabolite, and too difficult to find metabolite whose structure has changed significantly. On the other hand, in case of RI ADME study, the unchanged and its metabolites are tracked by RI, so we can evaluate quantitatively the total amount including metabolites by measuring radioactivity in sample. Therefore, the biggest benefit of RI ADME is that we can evaluate pharmacokinetics in more detail. In this slide, we will introduce novel labeling technology for nucleic acid called FT labeling. The middle is conceptual image of this technology. Red and blue shows target nucleic acid, and green is complementary strands that of target nucleic acid, including transfer unit. The detailed reaction scheme shows below. First step is hybridization, and thereafter functional transfer reaction have been occurred at adenine and cytosine's amine moiety, and transfer unit will be conjugated with target nucleic acid. The second step is labeling reaction using NABT4 and tritium labeled DNA and RNA can be synthesized. There are two other labeling technologies except of FT labeling. One is direct labeling and another is building up type labeling. The pros and cons summarized below. In direct labeling, low difficulty and low cost. However, the labeled position have been unspecified and the compound have low stability. In building up type labeling, we can label desired position and the compound have high stability. However, the method of synthesis is multi-step, so synthesis is high difficult and it takes high cost. In case of FT labeling, the labeled structure is different from target nucleic acid. The cons of two labeling technology can be overcome by FT labeling technology. For FT labeling, two ASO named LXA3945 and LXA8140 were provided by Luxana Biotech, and these ASO introducing modified nucleic acid called AMNA and scpBNA, highlighted by red and yellow, and blue is typical modified nucleic acid called MOE. Considering the location where the functional transfer reaction is likely to occur, we choose this position of adenine for FT labeling. The below table is the FT labeling result of two compounds. These results suggested that FT labeled nucleic acid was synthesized with good purity and enough amount of radioactivity for evaluation of ADME study. The upper figure is the radiochemical purity and mass data of both ASO, and the results show that the radiochemical purity of both ASO are good purity, and the accuracy of mass detect within 5 milldalton. The below table shows the ratio of tritiated water in urine sample. The production of the tritiated water shows low and suggesting that the FT labeled ASO have high stability in biological samples. Next, we conducted pharmacokinetics studies using FT labeled ASO. This slide shows study overview. We used two FT labeled test articles and the dosing decided based on previous study. At 8 weeks, BL6 mice were administered subcutaneously, and exclusion, distribution, metabolite analysis study were conducted. In exclusion study, urine, feces, and expired air were collected for 4 weeks, and radioactivity was measured by liquid scintillator counter. In distribution study, the section preparation was conducted, and the intensity of radioactivity was imaged by bath, and thereafter the concentration of radioactivity in designated tissues were quantified. In metabolite analysis study, the pooled urine was centrifuged and the supernatant was analyzed by LC-RAD Q-TOC mass system. This slide shows the exclusion study result. At 4 weeks after administration, the total excretion radioactivity was over 70%, and the main excretion route was urine in both ASO. The urinary excretion is generally known in LSO, and similar excretion characteristics were observed in both FT-labeled ASO. This slide shows the distribution study result. We conducted QWBA, named quantitative whole body autoradiography. In QWBA, we can obtain this autoradiograph image, and blue is low concentration, green is middle, and red shows high concentration. After administration, both FT-labeled ASO distributed rapidly to the whole body, and the river showed the highest radioactivity concentration, followed by the renal cortex. This is the radiochromatograms result in urine from 0 to 8 hour and from 0 to 72 sample. The profile of metabolites was similar for both FT-labeled ASOs. The FT-labeled ASOs metabolites could be quantified. Below table is a result of sample processing and column recovery, and the value was 90% over. Therefore, good analytical conditions have been developed. Based on the quantification result, the main metabolites were selected, and structure estimation have been conducted. At urine number 8 peaks in both FT-labeled compounds, based on the MS and MSMS data suggesting that these are the duplicated metabolites of unchanged. This is the last slide about summary and future prospect. In the RI synthesis, because of the small production of treated water, the FT-labeled nucleic acids seem to be stable enough for pharmacokinetic studies. In exclusion study, ASO is known to be gradually excreted mainly in urine as a full-length and short-chain forms, and both FT-labeled ASOs showed similar exclusion in the exclusion study. In distribution study, for QWA, both FT-labeled ASOs distributed rapidly to the whole body, and liver shows the highest radioactivity concentration, followed by the renal cortex. In metabolite analysis study, profiles of metabolite were similar for both FT-labeled ASOs in the urine, and FT-labeled ASOs metabolite could be evaluated. In future prospect, further evaluation of FT labeling will be conducted by comparing ADME data of approved nucleic acid. These data suggested that FT labeling technology is useful for pharmacokinetic study aimed at developing new nucleic acid drugs. Thank you for attention, and if you're interested in our RI ADME service, please feel free to contact us.
So to wrap up today's presentation, I'd like to talk about some important points. One is that BioAgilytix is a one-stop bioanalytical service provider for pharmacokinetics for all modalities, including oligonucleotides. Our ADME support is complemented by our partnership with Sekisui Medical and permits us to provide definitive radiolabeled ADME assays. We leverage all of our wide array of platforms for PK, immunogenicity, biomarker, and CMC bioanalytical testing to provide comprehensive support for your drug development programs. Our global locations allow us to serve studies around the world with local resources supported by over 600 scientists' expertise worldwide. If you have questions, please feel free to reach out to us and we'd be happy to answer them. Thank you very much for your time, and we look forward to hearing from you.